Hi, everyone. Welcome to today's webinar on MDDB and MemProtMD. I'm still seeing few people joining in, so maybe let's wait 2 minutes and then we can start the webinar. I think the number has stabilized now, so we can start. Hi, everyone. I am Preeti Chaudhary. I am working as a senior bioinformatician and project lead at PDBE at EMBL EBI. And we are one of the partners of MDDB project. And this exciting project is led by IRB Barcelona. We also have Barcelona Supercomputing, Nostrum Bio Discovery, Royal Institute of Technology, KTH, University of Oxford, and CCAM as our collaborators. And just to give you a brief introduction about the project, so MDDB is a collaborative initiative where we are bringing some of the leading institutes in Europe to transform handling of MD data and to facilitate sharing within the scientific community. MDDB project, it's aiming to build a unified database that compiles and organizes all the data generated by molecular dynamics simulation. And the idea is to set up a global repository for biosimulations data, just like we have Protein Data Bank, which serves as a global repository for experimentally determined macromolecular 3D structures. We would like to have something similar, kind of a global repository for MD simulations. And with these webinar series, MDDB aims to engage the MD community, and also it's an exciting opportunity for us to give you the sneak peek of all the exciting work and collaborations happening in this project. And today, this is our fourth such webinar of this series, and we will be discussing MemProtMD, a database containing over 7,000 molecular dynamic simulations of individual membrane proteins embedded in lipid bilayer. And speakers will introduce the feature of the database and discuss how it has been integrated in MDDB project. And without a further ado, it's my pleasure to introduce our speakers today. First, we have Dr. Charlotte Lynch, computational scientist specializing in molecular dynamics simulations of biomolecular systems, machine learning, and material modeling. She is currently a postdoc research associate at University of Oxford, where she is contributing to develop molecular dynamics database. Joining her is Professor Phil Stansfeld, a leading expert in computational biology. His research focuses on using advanced computational methods, particularly multi-scale molecular dynamic simulations, to investigate membrane protein structures. He's currently working as a professor in computational biochemistry at University of Warwick, where his research group is working on bridging structural biology and computational modeling to deepen our understanding of membrane proteins. So please join me in welcoming Dr. Charlotte Lynch and Professor Phil Stansfeld. And with this, I would like to give the floor to Charlotte to tell us more about MemProtMD. Charlotte, over to you.
Thank you very much, Preeti, for the introduction. So yeah, I'm Charlotte, I'm a postdoc at Oxford, and I'm going to talk about the MemProtMD database and how that links into the MDDB project. So just to sort of add just a little bit more detail on the Molecular Dynamics Databank project, which Preeti's also very nicely introduced for us. So our objectives in our international project are to design a database for the dissemination of molecular dynamics data according to FAIR principles, to promote the sharing of molecular dynamics data, to establish best practices for generating and analyzing that data, and to also integrate existing specialized molecular dynamics databases together more. And so the idea here is that we know that molecular dynamics simulations are becoming increasingly important as a tool for looking at biomolecular systems. And users these days are generating terabytes of data. And there's a sort of a drive within the community that it would be really good to share this data rather than sort of once you've finished with your own analyses to say delete some of the larger files, of course, keeping the key ones where you can still regenerate. But instead of having to delete those, it'd be nice if we could store them. And then that would mean that other people could look at those trajectories and use them for different analyses. And also it would mean that our data is more reproducible. And so if you're interested in more of our work and our news, then there's the website here that you can look at. So fair data, what this kind of means, broadly speaking, is that it should be findable. So we should have some sort of persistent, unique identifier for that data, and that should be easy to find for humans and also as well for computers. Accessible means that you should be able to access it via sort of standardized means. Interoperable means that it should be able to work with other different sorts of workflows or applications and so on. So you should be able to work with it, basically. And reusable means that you should have a well-described set of metadata so that you could replicate it. And the benefits as well for this fair data is that you can help with the reproducibility of simulations. You can also then generate new science from it. So you could do extra analyses on things that, say, the trajectories weren't originally run to look for. They could also serve as data sets for machine learning, for force field optimization. And also, if you don't have to rerun simulations, then that's helping with efficiency, energy efficiency as well. It can also help as a sort to serve as a bank of best research practices as well for molecular dynamics simulations, and also to help foster further collaboration as well between groups. And so the MDDB project, we started off with a whole set of different sort of existing test cases, and they are spanning a wide range of different types of biomolecular systems. So we have nucleic acids, central nervous system, proteins, the COVID-19 database, toxicity-related proteins, and then finally the MemProtMD database and the membrane protein one, which is the one that I'm involved in. And the idea as well is that all of these different databases, we're using them as means for testing and for designing the overall MDDB infrastructure. So more about MemProtMD. So there's a web interface, which you can access it all via here. And this is a project that's been running now for about a decade or so. And it has a web interface, which was designed by Tom Newport. And if you were to go onto the website, then you'd see a web interface that looks a bit like this. And so what this database is a whole set of simulations, or the outputs from the simulations of membrane proteins, which are embedded in lipid bilayers, and they're embedded in lipid bilayers using coarse-grained molecular dynamics simulations. And the reason why this came about is that, so to begin with, the crystal structures aren't given in their lipid environment, and so you're missing out on looking at the different interactions between the protein and the lipids. And also, if you were to run your own molecular dynamics simulations of membrane proteins, then embedding that protein into a lipid bilayer is non-trivial. And so this website gives you a whole set of output files where you have the protein embedded in a DPPC bilayer for you, if you want to use that further. So now, we've got more than 7,000 proteins in here, and this is this whole sort of pipeline and work is from Thomas Newport, Mark Sansom, and Phil Stansfeld, who's going to talk after me. So the web interface, you can see that you can have a look at sort of the latest simulations if you want. You can also search for simulations or proteins by name or by their PDB code. And then there's also groups of different types of membrane proteins that you could then have a look at. So then if you were to look at one particular protein in more detail, what you would see is an image of the protein embedded in the bilayer, and then some details as well about the original structure, where to find the original paper, and so on. You can also see what other databases that the protein is related to, other structural databases. So there are links to external collections, such as the Protein Data Bank, MemProtKB, Membrane Proteins of Known Function, the Transporter Classification Database, Gene Ontology, and PFAM. And so the simulation that's sort of going on under the hood to get to this sort of stage of showing you this on the website, what we're doing here is a set of coarse grain simulations. So each bead, which are all these little dots here, are representing several atoms. And the green and sort of greeny bluey beads here are representing parts of the lipids, and the yellow and the pink are representing parts of the protein. And the simulation starts off with a set of disordered lipids. So that's what we can see here. And then as the simulation runs, very, very quickly assembles into a bilayer. And this full simulation then runs for 1 microsecond. And the final frame is then converted back into an atomistic representation. And then we do a set of analyses on the last 800 nanoseconds. So we're making sure that this system, really making sure that this bilayer has fully assembled and it's all equilibrated by the time we do the analyses. And on the individual pages as well of the website, you can get a set of coordinate files for the final output, so coarse-grained or atomistic, and a set of simulation files so that you could then run your own simulation starting from this final output frame if you wanted to. And the pipeline is described in more detail here in this paper. So on each of the web pages for the different structures, there's also a set of analysis tools that you can have a look at. So we have outputs for the distortion of the lipid bilayer. So red here is representing bilayer thinning with respect to bulk, and blue is representing bilayer thickening with respect to bulk. We also have a set of contact analyses. So this is a contact is taken to being within 6 angstrom. And so we have contact analyses for the residues with respect to the lipid head groups, the lipid acyl tails, and also the solvent. And the darker colours represent longer contacts and higher average occupancy over the simulation. You can also click on the individual chains and have a look at those in more detail. And it will tell you whether it's membrane spanning or touching, or if there's no interaction. You can also see a breakdown of, with respect to each of the different residues, you can look at its contact with the different parts of the lipid, or the contact analysis for the solvent or the heads or the tails, and also the displacement from the bilayer center as well. So lots of things to look at there. You can also select the chains and have a look at the overall secondary structure, and then you can superimpose on that secondary structure the contact analysis. So you can look at the contacts with respect to the lipid head groups, the tails, the solvent, and the upper and lower leaflets of the bilayer. And so just to explain a little bit more what's going on under the hood in terms of the pipeline. So this is a fully automated pipeline. And what the pipeline does is that it checks the protein data bank for new structures. The simulation is set up. The protein is sort of oriented and it's predicted which parts will span the membrane. Then that whole system is energy minimized and it's sent over to our local cluster. The 1 microsecond coarse grain simulation is run. That's then brought back onto our local partition. There's some centering and orienting of the simulation that's done, and the final frame is converted back into atomistic. And then all the analysis that I've just discussed, that's done. The images are generated, and all of that information is then uploaded onto the web server. And so finally, we'd like to ask the audience, what sorts of things would you like to see in a molecular dynamics database, generally or for membrane proteins? So you can put suggestions in the chat for us and we'd be happy to hear them. Things that we've been sort of possibilities are things like looking at different ligands, mixed lipid systems, ion channel annotation. Of course, things like how would you want to go about uploading your data in the first place, and other links between databases. And then just to say thank you very much to everybody who I work with on this project. And I'm happy to take questions at the end of the session, so I'll pass over to Phil now. Thank you.
If I could just check with one of the other moderators that everything appears okay on their screens.
Yeah, looks good, Phil.
Okay, fantastic. Okay, thanks very much, Charlotte, for giving an overview of MemProtMD and also the MDDB as well. And what I want to do is to give you a bit of insight about where we are currently with MemProtMD, a bit of an overview from sort of previous work within the research group, but also what we've done with respect to MemProtMD and how we've used the overall workflow pipeline to then work on a variety of different systems. And beyond that, what I also want to talk you through are some recent pieces of work that's been going on within my research group to try and speed up the process of orienting membrane protein structures, but also building systems around them, building a bilayer, both a sort of plain vanilla phosphatidylcholine bilayer, let's say, or more mixed lipid systems as well, as you've seen from various other research groups worldwide. Charlotte gave a really great introduction to MemProtMD in terms of how its overall setup is, taking a PDB structure, generally on a Wednesday when they're released from the protein data bank, and then we turn it into a Martini representation, add lipids and solvent, and then you do it via a self-assembly process, receive a membrane protein structure within its lipid bilayer. We can then convert that system back to atomic level detail, and I'll talk you through that process in a couple of slides' time. And then also, as Charlotte suggested, we've got various different analyses that come out from those simulations at a coarse-grained level. So it's been said about 7,000 or so membrane protein structures. I think we're currently now in the 8,000s. And if I compare to when we published our paper only 6 years ago now, to where we were then in terms of thinking about the number of structures, there's a great deal more membrane protein structures for us to work with. And this has been enhanced even further by the developments of AlphaFold and other similar tools. And therefore, we have pretty much every single membrane protein structure, at least a prediction, to build a bilayer around those protein structures. So from that perspective, many users have ultimately wanted to use our workflow and pipeline to build bilayers around protein structures. So over the course of the pandemic, AlphaFold came out, AlphaFold 2, and they very generously, alongside the CollabFold team, set up Google Collab notebooks, which you could run and then fold your protein structures based on sequence alone. And so what I thought would be quite rather useful is whether we could use a Google Collab notebook to actually build membrane protein structures and bilayers. And so both using the self-assembly approach, but also methods from Suet-Jan Marink's group in SANE, we could then build bilayers around these protein structures using the workflow. And so if you want to do this, this particular system will take protein structures from the protein data bank, from the AlphaFold database, from the ESM Atlas databases, but also upload your own protein structures. And then you can build your own custom-made lipid bilayer around this protein structure using this set of protocols. So you don't need your own workstation for this. Ultimately, all of this is done within a browser. And in part, I did this for the community, but also it's very useful from a point of view of teaching as well, getting people familiar with membrane proteins, but also membrane protein simulations as well. And so even if you don't simulate using this overall workflow, what it ultimately gives you is your protein structure embedded within a lipid bilayer that you could potentially use as a figure for a publication. So various workflows are available, both for membrane proteins, but also peripheral membrane proteins, so both integral membrane proteins and those that bind at the interface. And hopefully this is documented enough. Go to the GitHub and fire up any of these collabs to then set away your membrane protein simulation. I'm happy to take any questions and comments, and certainly if anything's not working currently, please do let me know. So that was sort of giving you an overview and flavor for what we can do in an automated fashion, being able to simulate these membrane protein structures directly from the protein data bank or otherwise other databases around the world. But ultimately, what we also want to be able to do is to work with experimental colleagues who've used either X-ray crystallography, NMR, EM, to solve their membrane protein structure, and then prior to publication, build in some molecular dynamics simulations to try and interrogate the protein and understand further function of that protein. And so for several years now, and dating back probably longer than 2012, but 2012 is one of my first example I like to give on this slide, where we've taken a membrane protein structure and collaborated with our experimental colleagues to hopefully provide further function and details to these proteins. So most of these proteins are bacterial in nature. They're found in the bacterial cell envelope. And most of these proteins are also involved in either transport processes or otherwise enzymes. And sorry, there's a bit of a mistake here on the side. So we have got a publication just out in Nature Comms of this enzyme PIME in collaboration with Filippo Mancia's group, who we've collaborated on a number of these recent publications. So in all cases, what we're doing is taking this frozen snapshot of a protein structure using high-performance computing, including the MemProtMD workflow, to then allow molecular dynamics simulations of these proteins within bilayers, but also interacting with their native substrate molecules or otherwise potentially inhibitors as well. And so the modus operandi within my research group is really to study and focus on these bacterial biogenesis processes, how things are made within the cell envelope. And with the respect of understanding how these processes work, we can have a better idea of generating novel antibiotics. But hopefully you may be sort of also thinking, well, why do we care about lipids? I've talked a lot about membranes and membrane proteins, but I haven't really given you any explicit examples of why they're important. So what I just want to do on this slide is give you 4 examples from some of the work that I've just talked about on the previous slide. And these were all work by Robin Corey when he was a postdoc within my research group. Robin is now a group leader down at Bristol University. And so Robin ran a series of molecular dynamics simulations in coarse grain, in this case, to study the interactions of lipids with membrane protein structures. And so what Robin was able to do was to identify that lipids are really important, not only for solubilizing these membrane proteins, but for this ABC transporter, which has a central cavity within it. The lipids actually served as a bit of a plug. And so the plug prevented both the transport of the substrate, but also ions and water from leaking across the protein when it was in this open configuration and not transporting its substrate sugar molecules. He was also able to show scrambling of lipids from one leaflet to another. And this was one of the very first examples we used within our MemProtMD paper from about 10 years ago, where we showed scrambling of lipids within a fungal TMEM16 homologue of this, which is 16K, which was published a couple of years ago now. And so this is not an ion channel, unlike some of the other homologues of the TMEM16 family. It actually allows lipids to move from one leaflet to the other. And what you can hopefully see within the image are the spheres representing the phosphate groups of the lipid membrane. And this enables a sort of view of the pathway across the membrane by this scramblase molecule. And so the molecules can move in both directions within the membrane. And so I was recently at the Biophysical Society meeting. It is Biophysics Week this week. And so at that particular meeting, there was a lot of really cool simulations, in particular from Ben Corey's group, showing a lot of simulations of scrambling lipids across membranes. Another example is where lipids bind at the interface between subunits, and this was a collaboration with Simon Newstead's group. And so what Simon was able to identify was that within the experimental density, they could have an idea of lipid-like molecules bound at the interface, but he would ideally wanted to characterize which lipids they were. And Robin was able to identify that these were phosphatidylethanolamine lipids binding at the interface and therefore stabilizing the scaffold between these two proteins so that you got a stronger interface, a stronger dimer interface, not just protein-protein interactions, but also protein-lipid-protein interactions at that interface. And then one final example with Inga Henaut's group across at Frankfurt, we were able to simulate their Kim-A protein. Inga came to us ultimately saying that they weren't certain that the structure that they resolved was actually the correct conformation of their protein. And the feeling was that the two domains were a bit too close to one another, and hopefully you can see the V-like shape of the membrane previously observed within the simulation. Over the course of the simulation, we ultimately get to this planar lipid bilayer state, where lipids are able to penetrate in between the two subunits. And it seemed to be a virtue, therefore, of the experimental EM method that they were using, that they weren't able to capture the lipids suitably between that interface, and therefore they didn't get a stabilized scaffold. So ultimately, what I would say here is that the membrane itself is really important. It's really important for overall structure in membrane proteins so that they're in the right conformation. It bridges the interface between subunits. It can move from one leaflet to another through channels formed by membrane proteins, and the lipids can also plug pores within proteins. Lipids are also really important from a biogenesis point of view because there are also a series of enzymes within the membrane that use lipids and lipid-like molecules as substrates to do their processing. And so for me, they're one of the most important molecules within life. Now, I talked to you previously about going from a coarse grain representation, this reduced representation where 4 atomic atoms, usually non-hydrogen atoms, are mapped to a single bead in the coarse grain representation. What we want to be able to do is not just be able to go from atomistic to coarse grain, but also go back. And so many years ago, I developed a method called CG2AT, which took fragments of lipids and glued them back together to then allow you to go from this coarse grain representation shown in spheres, then back to a full atomic level detail. And so Owen Vickery, who was a postdoc in my group, was able to develop an improved version of CG2AT, which really refactored it so it was then in Python 3. And so that methodology is now available online in my GitHub, both a full version called CG2AT, but also a light version that I've recently put out that is a sort of simpler version that simplifies the conversions so that we only consider Martini 3 being converted to Charm 36. The overall method works not just for lipids, it also works for proteins, and it also works for post-translational modifications in some instances as well. And so this is a fundamental part of the MemProtMD pipeline and really why I highlight it at this stage. From an analysis point of view, there are a couple of other packages that were developed in collaboration with Mark Sansom's group. And so Wanling Song, who was a postdoc within Mark's group, and in collaboration with me, with Wellcome funding, was able to generate this PyLipID package, which allows the analysis of specific lipid interactions with a membrane protein. So let's say you have a mixed lipid system. You want to be able to identify which lipids bind to which points on the protein structure, and PyLipID will give this analysis, both identifying the lipids within the annular shell of the membrane protein, but also highlighting the residues that are in direct contact with the protein structure. And Bertie Ansell, who was a DPhil student within Mark's group, but also working with me, was able to identify a methodology called lipidens, where you could characterize the overall surface of a membrane protein and take experimental EM density to then work out regions that were undefined in terms of which regions around the protein weren't characterized already as protein. And so this sort of tube-like density could often be described as lipids, but it was uncertain which lipid species was binding at each and every one of these points. And so we could use these coarse grain molecular dynamics simulations with the Martini force fields developed by Sivert Marink's group, and then identify which lipid was potentially binding at each and every one of these sites. And so what this ultimately shows is a sort of close agreement between experimental data for protein-lipid interactions, but also relates that then to this simulation-based approach. And so from this data, we can then characterize which lipid is most likely to bind at the binding site that was identified in the EM density. I just want to move on now to talk about a specific case study that was done by Rob and Corey using PyLipID, in this case to identify where cardiolipin binds to a range of different membrane protein structures. So this is cardiolipin. It's a unique lipid. It has 4 lipid tails, 2 phosphate groups that are joined by a glycerol group. And in effect, they're 2 phosphatidyl glycerol molecules connected together. And so these lipids are really important both within mitochondria, allowing some curvature and interactions with membrane proteins within the cristae, but they're also found readily within bacteria at about 10% concentration. And so therefore we were interested to know whether we could identify lipid binding sites to a wide range of membrane protein structures from E. coli for which we had experimental structures. So from this data, we were trying to work out, is there a common binding site for cardiolipin? Which residues within these proteins are integral to binding? And can we therefore, based on this, devise a set of rules for binding of cardiolipin to these membrane protein structures? And so from this, and summarizing quite a lot of work, Robin was able to convert this to a coarse grain system, but then identify from these simulations, probably not surprisingly, that arginine and lysine, the positively charged amino acids, predominantly interacted with the negatively charged phosphates of cardiolipin. I think that was pretty much a given, but this is something that then came from his data set. He also identified that generally some small residues, serine and glycine, also bound in close proximity to this region. And so that also captures this GL0 bead that connects the two phosphate groups. The tails were predominantly interacting with those hydrophobic residues, but it was quite nice to be able to characterize that. And then what we found is that you again got hydrophobic residues interacting with the glycerol groups. Arginine was there too, but also some aromatic groups that are usually found to anchor the membrane protein with respect to the lipid head group region. And so Robin was also able to identify that most of the interactions, or a significant number of interactions, were happening in the inner leaflet of the membrane. And that suggested, therefore, potentially there was an asymmetric distribution of cardiolipin across the membrane, at least in its interactions with those membrane proteins. And this is also true for the distribution of arginine and lysine residues that form contacts with the lipids. And so many of you will know this is the positive inside rule, where arginine and lysine residues are predominantly expressed on the inner leaflet of a membrane protein, certainly within bacteria. And so there seemed to be a commonality between the cardiolipin interactions and the arginine and lysine interactions. There's a case of a chicken and egg question here. Are we getting more cardiolipin interactions just because there are arginine and lysine residues present? Or are we finding more arginine and lysine residues present so that they can then interact with the cardiolipin within the inner leaflet, which has a greater distribution within the inner leaflet? And I think that's still to be thoroughly determined. So from all the PI-lipid analysis, we had about 900 different analyses that we could then study in more detail and characterize which residues were likely to be interacting. And I'm just going to summarize this in this sort of figure. So what we were able to identify was that the majority of cardiolipin interaction sites were on the inner leaflet of the membrane. And there were generally two basic residues, arginine or lysine, that were about 0.8 nanometers from each other. And they were interacting with the phosphate groups of the cardiolipin. And they were generally found on looking across the membrane within about 0.3 nanometers of one another. They about 1.8 from the center of the bilayer. And there was generally one of these small polar residues at the interface between the two phosphate groups. We also saw a variety of aromatic residues coordinating the cardiolipin residues, therefore engaging with these lipids within the membrane. And so what we determined that this would be a Cori cluster, an interaction site for cardiolipin with membrane protein structures. And so recently we've taken this on and in collaboration with Michael Landra's group across in Sweden, we were able to identify a binding site for cardiolipin to an actual designed protein. So this protein is called rocket on the basis of the structure of the particular protein. It looks a lot like a rocket. This was designed by David Baker's group. And perhaps this was one of the reasons why David Baker won the Nobel Prize last year for chemistry. And so this design protein is a membrane protein. It rather nicely sits within a membrane using our MemProtMD pipeline. And in the simulations, we were able to identify a cardiolipin binding site to these proteins, which was in good agreement with the experimental data from Michael's group. We could mutate this binding site, and that would then disrupt the binding of cardiolipin. And so the red regions of here on this particular plot shows key interactions between the cardiolipin and arginine and lysine residues around this rocket protein. And then if you mutate these arginine and lysine residues, you lose the binding probably unexpected. You're taking away the positive charges. That's going to reduce these negative charge interactions. But what's rather nice is you can use the likes of Chi and AlphaFold to then propose an interaction of these cardiolipin molecules with the membrane protein that agrees very well with what's we've previously shown through these molecular dynamics simulations. So if you want to read this in more detail, it's only just come out, I think, this week in eLife. So have a look there at this particular study. But just gives you an example, we can understand cardiolipin interactions using molecular dynamics simulations from known experimental structures from in vivo proteins, real world E. coli proteins, but we can also study this and design proteins that can directly bind cardiolipin to it. I'm going to change gears ever so slightly. I'm just conscious of time. And what I want to talk you through now is a new method that we've developed within the research group. In particular, pretty much all of the work shown here is by Matthias Parag, a PhD student within my research group. And so what Matthias has been developing is a method to hopefully speed up setting up membrane protein structures. And so one of the crucial aspects of setting up any system is to be able to orient the protein correctly within a membrane, but also then to take that orientation and directly then simulate that particular protein onwards. And so that's partly why we set up the MemProtMD workflow on the Google Collabs. But we thought it would be useful to set up and establish a new system so that we can do this all in one particular package. And so this package we call MemPro. It's membrane protein orientation. It takes a set of coordinates for a membrane protein. From that, creates a coarse grain model, extracts the surface, and then tries to align that surface with this potential, and therefore repositioning the protein with respect to the membrane, which you can then convert back to an atomistic and establish a bilayer around it. And so the data you get out from this is a sort of potential energy associated with the orientation of the protein. And so this is the particular orientation for this membrane protein within its bilayer. And so we don't just want to do this for single membrane proteins. We want to do this for more complex systems as well. If you want to give this a go yourself, please do take account of this QR code. I'm happy to share this with anybody if the QR code, for whatever reason, does not work. And so the overall idea is that you can orient your membrane protein and then using INSANE, which was developed by Cirque Vassanar in Sirit Yang Marink's group, and then build a membrane protein around it. And so we've got our own edited version of INSANE that takes our MemPro prediction and then establishes the overall system around it, ready for coarse grain simulation, but also conversion back to atomistic to then run atomistic and CHARM or AMBER simulations of these membrane proteins. And not only does this work to orient single membrane proteins within their bilayer, it also works on double membrane systems. And as I said in the outset, we're very much interested in cell envelopes of bacteria. And many bacteria are didomes. They have two membranes, both an outer membrane and an inner membrane. And what I just show you here are a series of efflux pumps for which we can use MemPro to orient not only the inner membrane system, but also the outer membrane protein system above. And so beyond this, we can then build both the outer membrane and the inner membrane and establish a full molecular system around it. And we have a final system that is enabling the prediction of where we believe the peptidoglycan layer to sit. And so this peptidoglycan layer is the cell wall. It's the archetypal, I guess, the Achilles heel in many ways for bacteria, because we can target the penicillin binding proteins with beta-lactam antibiotics such as penicillin, and therefore stop the growth of the cell wall and cause cell lysis. But from our perspective, we just want a method to be able to model this. And so using parameters that have been generated elsewhere, we can then build a peptidoglycan system around this, both multiple layers in the Z dimension but also in the X and Y as well. And so this is in coarse grain at present, but you may have seen that Saima Khalid's group has also developed an atomistic representation or recent atomistic representation of peptidoglycan and a method for building that. And so therefore there will be multiple ways to build peptidoglycan around these protein structures. So not only does this work for things like periplasm spanning proteins, it also works for gap junctions where you may have connexins, a couple of connexins on top of one another, or maybe a stacked pair of aquaporins within the lens. And so this will work for many double membrane systems where you have two bilayers stacked on top of each other. The method also works for curved membranes as well. And I often tell various colleagues that membranes shouldn't just be drawn as two straight lines, two dashed straight lines, because they're highly complex and highly complicated. And in many cases will undergo large-scale conformational flexibility. Such as these piezo channels cause an enormous amount of curvature. And so what we wanted to be able to do is to predict this curvature, but also be able to build the systems around these curved membrane systems. Bar domains have been known for several years, and there's been some fantastic simulations from Greg Voth's group over the years, looking at these bar domain interactions with membranes. And several simulations as well done on these mechanosensitive channels, again, ones that respond to pull and stretching of the membrane to then open the ion channel within. And so this may involve a level of curvature that's shown here. Alternatively, it may be a more local curvature, and I'll come on to that in a second. So as I've said, you can use this INSANE for MemPro methodology, which is a development of INSANE, to build these curved systems around the protein structure, and therefore hopefully get a more accurate representation of what the bilayer should be with respect to these proteins. And furthermore, we can build micelles and try and represent and build experimentally used methods to capture membrane protein structures. So not only micelles, but also nanodiscs, but also SMA nanodiscs, SMALPs as well. And one thing I was just coming on to there was this curved MemPro prediction of this mechanosensitive channel. And what we saw within MemProtMD in our database entry was that this does cause quite a bit of curvature of the membrane around the protein structure. And so what we decided to do is develop a particular method that also takes into account distortions of the membrane around these proteins. And so this is a new version called MemProd, building on MemPro's orientation method to also capture distortions of the membrane around these proteins. So not just needing simulations to capture these distortions, but being able to predict ab initio the distortions around these protein structures. And so I just want to finish with a couple of examples, therefore. An example of a protein that we worked on with Filippo Mancia's group called WAL. This particular protein binds a molecule called lipopolysaccharide, which has shorter tails within its lipid membrane than conventional lipids. And so we were able to identify within our molecular dynamics simulations a thinning of the membrane shown on the right-hand side. And this was equivalently seen within the MemProtMD database and the data set. This thinning of the membrane was where the lipopolysaccharide was binding. And with this MemProd prediction, which takes about 5 minutes to run, we can get something similar in terms of its distortion around that particular binding site. Similarly, I talked about TAT-C, right, as that 2012 paper that we had with Ben Birks's group and Susan Lee's group. This also shows a thinning both within MemProtMD, but also within our MemProd prediction. And it's at this particular binding site where we believe the other subunits, TAT-A and TAT-B, bind to this protein. These are shorter transmembrane helices, and therefore there's a requirement for a thinning of the membrane, and that's captured by this distortion prediction. One final example, going back to scramblazers. Scramblazers can be open and closed. What the distortion methodology can also do is identify whether it's likely that the channel is going to be open or whether it's going to be in the closed configuration. And hopefully you can see this upper distortion is not captured within the closed state, whereas it is distorting quite distinctly down this scramble channel that I showed you in an earlier simulation by Rob and Corey. Okay, I'll stop there. What I've talked to you about in the past 30 minutes is our overall pipeline for MemProtMD, building on what Charlotte told you originally. I've shown you how you can use our molecular simulations in terms of those coarse grain molecular simulations to identify cardiolipin lipid binding sites. And then I've moved on to tell you about our new methodology, MemPro, developed by Matthias Parag, for orienting both single membrane proteins, double membrane systems, but also these more complicated curve systems. And then finally, a methodology for predicting distortion around membrane proteins called MemProd. All of this is found within our GitHub, at least at the moment in Matthias's GitHub. And if you can't gain access to that, please do let me know and I'll happily share it with you. Many people to thank, too many people for now. But ultimately, the key work I talked to you about was initially initiated by Tom Newport, a DPhil student within my group, to set up the MemProtMD database. I've talked to you about Rob and Corey's various simulations, Owen Vickery's CG2AT, and then towards the end, I talked to you about Matthias Parag's methods for predicting and orienting membrane protein structures. All that's left for me to do is really to thank you for your time and attention this afternoon. I'll be more than happy to take some questions after this session.
Thank you, Phil, and thank you, Charlotte, for giving us the overview of MemProtMD, all the nice use cases and flavor of what, we can get such nice biological insights from the simulations, all your nice tools. Thanks for that. And with that, we will now open the floor for questions. Just a reminder for everyone, you can use the Q&A box at the lower panel of your Zoom next to the chat to type the questions. We already have few questions, so we can start. The first question is from Valentine. He says, "Hi, thank you for the presentation. Could you please explain what structural changes are normal to observe from experimental structures to the one embedded in a membrane? Also, maybe a bit naive, but are experimental structures necessarily determined in aqueous media?"
So I mean, I happily take that. So I guess from the point of view of the coarse grain simulations that we run, these generally will have an elastic network imposed upon them. So therefore, we try our best to retain the starting structure throughout our simulations. So within the MemProtMD pipeline, we're not looking for necessarily for conformational changes within the protein structure. So we keep that as close as possible to the starting conformation. And so ultimately, these are membrane proteins. In terms of the aqueous media, I guess we'd still require solvents of some nature, and these are usually detergents that would then be surrounding the membrane protein to therefore interact with the hydrophobic region of the bilayer, or what the membrane protein would be interacting with. This could be otherwise nanodiscs that are used where you actually do capture the lipid membrane around the protein structure. But yes, you're right. You need to extract it from a membrane in many ways, its native membrane, to then have it in an aqueous media around it. But usually it's an extraction from the sort of native state.
Thanks, Phil. The next question is from Kian Carlo. I mean, you touched about solvent. So he's basically asking, I think, the technical details. How do you represent the solvent and what about the hydrogen bonds?
Yeah, so I guess the solvent itself is represented using the Martini water which is a reduced representation of true solvent. And certainly Charlotte probably can talk at length about what solvent we ideally should be using for any of our simulations. But yeah, maybe I won't go on to that. And Charlotte, I don't know whether you want to add anything at this point.
Just to reiterate, at the moment we're using Martini for it.
Thank you. The next question is from Lakshmi. What features does MemProtMD offer additionally as compared to Charm GUI?
I mean, let's be frank, Charm GUI is amazing. Wampa Lim and his team has done an absolutely wonderful job with Charm GUI, and I can't necessarily say that we offer anything new or different. We do what we ultimately have is an assembly of all these entries for membrane proteins that you could just access. So if you're interested, has that particular membrane protein got a bilayer around it already? Having been to the PDB, you would be able to directly download that protein structure with a bilayer around it. But in many ways, MemProtMD provides something different from what Charm GUI does. But equally, I would encourage you to use both MemProtMD and Charm GUI because I personally, I think Charm GUI is a fantastic resource for the community.
And also to be clear as well that MemProtMD does DPPC. So we haven't got other lipids in there.
Thank you. The next question is, I'm going to take two questions together because they are sort of related from Matthew and Lucy. They are talking about how we are going to integrate MDDB and MemProtMD, and are the analysis will be transferred to MDDB? If not, how MemProtMD and MDDB will cooperate or interoperate?
We're sort of thinking about using sort of common analyses within the MDDB framework and MemProtMD. One of the differences at the moment is that, so with MemProtMD, you can't upload your own simulations on there, whereas MDDB is going to be uploading your own simulations. And then we're thinking about different ways to either look at, say, see whether you've got the same systems in both MDDB and MemProtMD, and yeah, whether there will be similar analyses, swapping annotations. So there's a lot that we're thinking about doing there.
Right, thanks, Charlotte. The next question is from Bernadette. Thank you guys for your talk. Great work. What system sizes are you currently able to investigate? I'm thinking about DLP1 interacting with mitochondrial membrane.
You've gone away from my expertise. I don't know necessarily what DLP1 looks like, how many atoms it contains, and we can generally do pretty large systems, certainly of the order between 100,000 all the way up to over millions of atoms. And you'll have seen some fantastic talks, I'm sure, over the years of whole virus systems. Whether, I don't know, I can't comment with that. I would need to go and do my homework and find out what DLP1 looks like, but we try and do as big as possible.
Great. Thanks, Phil and Charlotte. I don't see any other questions in the chat box or in the Q&A box. So maybe we do have one last one. Maybe we can take this one. Do analysis like lipid or lipid ensemble also work for spherical systems, such as vesicles containing a lot of proteins?
I think they should, but I haven't actually tried it myself. But yeah, in terms of interactions, it's just pairwise, so that should be possible whether it's in a planar system or in a vesicle configuration. I don't see any reason why that would be an issue. It might take a while if you're looking at sizes of systems like a vesicle. There was another question I saw pop up, which was about hydrogen bonds in Martini. We don't have explicit hydrogen bonds, but there are information baked into the overall parameters that try to capture hydrogen bonds. It's just it's not necessarily that non-bonded interaction. It's not explicit.
Great. Thanks, Phil and Charlotte. Before we close the session, I just want to share with the participants the upcoming MDDB events. So the next webinar, the fifth and the last one of the series, is about DynaRepo, data set for conformational dynamics and allosteric annotation. And we also have an upcoming workshop, When Data Science Meets Molecular Dynamics. It's at CCAM in Lausanne. And for this, the registrations are open for both on-site and remote attendance. So please go have a look. And for all the upcoming events, please keep an eye on our MDDB website events page. With that, thank you for joining, everyone. Thanks for joining. And I would like to close today's session. Thanks, Phil and Charlotte.
Thanks, Preeti.
Thank you.
Thanks all for coming. Bye for now.
Bye.